Thank you. Okay, hello everybody. So my name is Petar Lukanov, and I'm a Marie Curie PhD student in Toulouse, France. And I'm working in a group of Dr. Emmanuel Plau in Toulouse. So we are part of one European project named CarBio, especially for bioapplication of carbon nanotubes for cancer treatments. I would like to thank for organization committee that I received my grant, and that's why I'm here, and I can present my work in this nice big conference. Thank you very much for the GDR grant. My talk today is filling of double carbon nanotubes with ion precursor in solution. First, I would like to say a few words about synthesis of double carbon nanotubes in our laboratory in Toulouse. The second place, I would like to explain a few things about the purification and opening of double carbon nanotubes. And finally, filling of double carbon nanotubes from solutions. So synthesis, we use catalyst prepared by combustion synthesis and CCVD in these conditions. So as a characterization of our carbon nanotubes, we have about 90% of carbon content. And on these pictures, we can see a cross-section of our double carbon nanotubes. It's actually pictures of some bundle of double carbon nanotubes. And also, we can see model of the structure of double carbon nanotubes. The statistic that we have for our tubes, we can say that between 70 and 80% are double carbon nanotubes, and the rest are single wall carbon nanotubes and triple wall carbon nanotubes. Also, from this statistic, we can say that the inner diameter is about 1.3 nanometer, so small, and also external is about 2.6 nanometer. Opening and purification of the carbon nanotubes. For our target filling, before that we should open the ends of carbon nanotubes and to purify them because of bioapplication. Our colleagues in the project working in their application and functionalization, of course. So that's why we are trying to purify them with and open with two types of oxidation. With first one with trimolar nitric acid, and the second one with mixture of sulfuric acid and potassium permanganate. So the conditions for both of these agents are at 130 degrees we have experiments for 3, 6, 9, and 24 hours. On this picture, we can see a reflux setup which we use every day for oxidation. The results which we receive for carbon percent after oxidation with these two agents, we can conclude that after 3 hours of oxidation, we have better results of oxidation with a mixture of sulfuric acid and potassium permanganate. The situation after 24 hours is almost the same for these agents. We have the same oxidation. Also, we have results for molybdenum and cobalt quantity in our samples after oxidation. So for molybdenum, we can say that oxidation with trimolar nitric acid for different times, it's better, it's more efficient than oxidation with permanganate for the same hours. The situation with cobalt is almost the same, just the difference is quantity of cobalt after 3 hours and 6 hours. But finally, after 24 hours, we have big distribution of cobalt as the same as in molybdenum. Of course, these metals are coming from catalysts, so we are trying to dissolve them with this oxidation. Also, we have Raman spectra of the same samples, oxidation with trimolar nitric acid. This what we can see is the blue spectra. It's oxidation after 24 hours, and we can say that the defect in the structure after 24 hours of oxidation are much more than in oxidation after 3-6 and of the raw carbon nanotubes after 6 and after oxidation 6 hours and 9 hours. Of course, it corresponds with defects in the structure. Almost the same situation is with oxidation with potassium permanganate, but the intensity of the D band after 24 hours oxidation is almost half comparison with the peak of the spectra of oxidation after 24 hours with nitric acid. Also, we have experiment for loss, weight of loss. So the situation here is corresponds with the previous result. So we have lost about 70-80% of mass after oxidation with nitric acid, and comparison with the other oxidation with potassium permanganate, it's the same as after 3 hours and 24 hours. So another evidence that oxidation with trimolar nitric acid is more efficient than oxidation with potassium permanganate. Also, we provide some experiment for about functionalization of our tubes. This result could be interesting for our partners in the project who are trying to attach some bioengines on the surface of our carbon nanotubes, especially for bio application. In this one, we are not so interested of functionalization, but also we have samples of a sample after oxidation 3 hours with nitric acid, spectra B, and spectra corresponding with raw carbon nanotubes. So we can see very clear